Discovering biomarkers and pathways shared by Alzheimer's disease and Parkinson's disease to identify novel therapeutic targets. Abstract: Alzheimer's disease, AD, is one of the most disabling and burdensome health conditions worldwide and a leading neurodegenerative disease that results in severe dementia. Parkinson's disease, PD, is also a neurodegenerative disease, and literature suggests pathogenic links between AD and PD, but the molecular mechanisms that underlie this association between AD and PD are not well understood or have a limited understanding of the key molecular mechanisms that provoke neurodegeneration. To address this problem, we aim to identify common molecular biomarkers and pathways in PD and AD that are involved in the progression of these diseases and deliver clues to important pathological mechanisms. We have analyzed the microarray gene expression transcriptomic datasets from control, AD, and PD affected individuals. To obtain robust results, we have used combinatorial statistical methods to analyze the datasets. Based on standard statistical criteria, we have identified 111 upregulated genes overlapping between AD and PD, and at the same time we have identified 20 downregulated overlapping genes between AD and PD. Pathway and Gene Ontology, GO, analyses pointed out that these 111 upregulated and 20 downregulated common genes identified several altered molecular pathways and ontological pathways. Further protein-protein interactions, PPI, analysis revealed pathway hub proteins, EGFR, JAK2, MAPK11, EIF3B, WASL, BCL2L1, CDH1, MCM5, RAN, NCOA3, TBL1X, RARA, RHG12, NCOA2 and ESR2. Transcriptional components were then identified, and significant transcription factors, FOXC1, GATA2, YY1, TFAP2A, E2F1, FOXL1, NFIC, NFKB1, TP53, USF2 and CREB1 were identified. We have performed protein-drug interaction analysis to reveal drug interaction with proteins. Thus, we identified novel putative links between pathological processes between AD and PD, and possible gene and mechanistic expression links between them. Keywords: Alzheimer's disease, Parkinson's disease, biomarker signatures, differentially expressed genes, protein-protein interaction, protein-drug interactions. Introductions. Alzheimer's disease is a chronic progressive neurodegenerative disorder causing severe dementia and cognitive decline in elderly people. 1,2. As the number of Alzheimer's cases rises rapidly in an aging global population, the need to understand this puzzling disease is growing. 1. As the disease advances, symptoms can include problems with language, disorientation, mood swings, loss of motivation, and behavioral issues. The pathobiology of AD involves the formation of amyloid plaques and tangles in neurofibrils 3 which may break up neuron function. Parkinson's disease, PD, is a neurological disorder characterized by motor deficits as a result of the progressive degeneration of dopaminergic neurons 4. In 1817, James Parkinson described the core clinical features of the second most common age-related neurodegenerative disease after Alzheimer's disease, AD. 5. PD is the cause of the second highest number of deaths worldwide, and raised to 81.1 million incidents by 2040 is predicted. 6. After age 55, the risk of PD and AD increases, although it can occur at any age. Thus, the mortality and associated morbidity for AD and PD make a major global healthcare burden. 7. There is a trustworthy record for epidemiological and pathological links between AD and PD from population-based studies which revealed that PD is linked with AD and vice versa. 8, 9, 10, 11. Undoubtedly, both of them involve neurological damage, and there may be shared pathological mechanisms underlying both conditions. 12, 13. Various studies have tried to mark out genetic links between PD and AD, for example, studies of genome-wide association. These have identified common genetic components, mainly single nucleotide polymorphisms in PD and AD that point to at least some shared element in their pathogenesis. 14, 15, 16, but the significance of these is currently uncertain. Till now, common dysregulated molecular components in both diseases is very limited between AD and PD17. However, due to the availability of data, candidate biomarkers, such as differentially expressed genes, DEGs, pathways, and ontology have been identified using transcriptomic datasets from microarray studies 18. Therefore, to utilize microarray data, we have employed a systems biology approach to identify dysregulated genes and molecular pathways that are common to AD and PD. We have used findings from such analyses to integrate the differentially expressed genes, DEGs, in AD and PD tissues with interaction networks using the following approaches: A and protein-protein interactions, PPI, network of the proteins encoded by the common DEGs, 2. Identification of transcriptional components of the common DEGs, 3. Protein-drug interaction networks to screen potential drugs. This comprehensive bioinformatics pipeline thus allows us to explore possible critical pathway hub protein transcription factors, TFS, transcripts of interest, and to provide potential biomarker signatures useful for disease assessment and further biological research, which is shown in Figure 1. We can also use these insights to identify drug-binding partners to some of the hub protein that may indicate new therapies if the proteins prove to have important pathological influences on AD and PD. Figure 1, the overview of the integrative bioinformatics approach employed in the present study. Materials and methods. Identification of differential genes expression in AD and PD. Differential gene expression is important to understand the biological differences between healthy and diseased states. 19. The microarray gene expression high-throughput analysis datasets for AD and PD affected tissues were acquired from the NCBI-GEO database. 18. The AD dataset, GSC 28146, was microarray data, also Affymetrix U133+. 2.0 arrays, on RNA from SNAP frozen brain tissue where white matter tissue was extracted by laser capture methods to collect only CA1 hippocampal gray matter from 8 control and 22 AD subjects. The PD dataset, GSC 19587, was an analysis generated from affected brain areas of 12 post-mortem brains of PD patients and 10 control samples of unaffected brain tissue using Affymetrix U133A plus 2.0 arrays. The datasets were first analyzed in the bioconductor environment implemented in R to identify DEGs in the PD and AD data relative to their respective matched controls. 20. Firstly, the gene expression dataset was normalized by log2 transformation and combinatorial statistical methods using the lemma package in R, Kruskal-Wallis, and students' t-test in hypothesis testing, with Benjamini-Hochberg correction to control the false discovery rate. 21. A p-value less than 0.05 and absolute log2 fold change, FC, of 1 was regarded as threshold criteria for significant DEGs of interest. Pathways and Gene Ontology enrichment analysis. Gene overrepresentation analyses were performed to identify molecular pathways and gene ontology, GO, i.e., biological process, cellular component, and molecular functions, terms using Enrich R22. Network analysis is complementary to pathway analysis and can be used to show how key components of different pathways interact. Using a set of genes that are upregulated and downregulated under certain conditions, an enrichment analysis will find which GO terms are overrepresented, or underrepresented, using annotations for that gene set. An adjusted p-value less than 0.05 was considered important for all the enrichment analyses. Protein-protein interaction analysis. Proteins are the workhorses that facilitate most biological processes in a cell, including gene expression, cell growth, proliferation, nutrient uptake, morphology, motility, intercellular communication, and apoptosis. Protein-protein interaction is becoming one of the major objectives of system biology. It plays a key role in predicting the protein function of the target protein and drug ability of molecules. The majority of genes and proteins realize the resulting phenotype functions as a set of interactions. We used the string protein interactome database 23 to construct PPI networks of the proteins encoded by the identified DEGs using topological parameters degree greater than 15. Network analysis was performed using the Network Analyst online resource 24. Identification of transcriptional regulatory components. A regulator gene or regulatory component is a gene involved in controlling the expression of one or more other genes. Transcription factors are proteins possessing domains that bind to the DNA of promoter or enhancer regions of specific genes. We used Network Analyst 24 to identify regulatory TFS that regulate DEGs of interest at the transcriptional level using the Transfact database 25 by setting the topological parameter degree greater than 15. Protein-drug interactions exploration. Drug Bank Database, version 5.0, 26 was used to analyze the protein-drug interaction. This process was used to identify potential drugs for patients suffering from AD and PD. Network Analyst was used to perform analysis of protein-drug interactions. A. Results. Differentially expressed genes identification common to AD and PD. The gene expression microarray datasets of AD and PD were analyzed and using applied combinatorial statistical methods for both datasets and significant DEGs were identified. 111 upregulated common genes were identified between AD and PD from the number of 948 and 2165 upregulation genes from AD and PD respectively. 20 downregulated common genes were identified between AD and PD from the number of 584 and 735 downregulation genes from AD and PD, respectively, which are shown in Figure 2. B. Figure 2, up and downregulated genes between AD and PD. Pathways enrichment and gene ontology analysis. After identifying DEGs, the set of identified common genes was used to gene set enrichment analysis to identify molecular pathways and gene ontology, molecular function, biological process, and cellular component. We considered 3 pathway databases, KEG27, Reactome 28 and Wiki Pathway database, and we retrieved significant pathways by EnrichR which are significantly connected with DEGs of AD and PD as shown in Table 2. To obtain further insights into the molecular roles and biological significance, enriched common DEGs sets were processed by Go methods using EnrichR, which identifies related biological processes, molecular function, and cellular component which are shown in Table 3. The list of molecular pathways and gene ontology terms was then curated to include those terms with a p-value below 0.05. The gene expression regulation is the process by which expression of genes is controlled at the cell level at a particular time under a particular condition. At the transcriptional level, the initiation of gene transcription is regulated by transcriptional factors. We identified the significant TFS to find out the transcriptional regulatory components of the common DEGs between PD and AD. We identified 11 TFS that regulate DEGs common between AD and PD. The transcriptional regulatory components are FOXC1, GATA2, YY1, TFAP2A, E2F1, FOXL1, NFIC, NFKB1, TP53, USF2 and CREB1. Protein-drug interactions of common DEGs between AD and PD. We studied the protein-drug interactions analysis and found that ESR2 and NCOA2 proteins have known interactions with several characterized compounds, namely genistein, diethylstilbestrol, raloxifene, tamoxifen, estradiol, trilostane, estramostine, dehydroepiandrosterone, para-mercury-benzene sulfonic acid, methyltrianolone, afamoxifene, estriol, estrone sulfate, 17-methyl-17-alpha-dihydroquinone, 3,3-fluoro-4-hydroxyphenyl, 7-hydroxy-1. Naphthenitrile, 4, 2-amino-1-methyl-1-H-imidazo 4,5-B-pyridine 6-YL, phenol, tributylstanonyl, raloxifene core. Discussion. In the present work, we studied gene expression data of PD and AD patients. To identify genes dysregulated in both diseases that may be potential therapeutic targets and candidate disease biomarkers, we employed bioinformatics pipelines. The candidate biomarker genes were explored by using microarray studies of PD and AD. Our proposed approach commonly applied to identify interactions between complex diseases 20,21,22. We have analyzed these gene expression patterns seen in AD and PD patients, which revealed numerous important alterations. The Gene Ontology and Pathways analysis exposed a number of neurodegenerative disease-associated pathways. However, this integrative analysis has effectively identified novel key or hub proteins that are common to these two diseases. It suggests new lines of inquiry for studies, including identification of possible therapeutic intervention for new targets. Protein-protein interaction analysis is broadly used to access important disease-associated signaling molecules and pathways which may raise aspects of a disease. Thus, we perform a PPI analysis to identify significant hub proteins. We have identified 15 hub protein, EGFR, JAK2, MAPK11, EIF3B, WASL, BCL2L1, CDH1, MCM5, RAN, NCOA3, TBL1X, RARA, RHG12, NCOA2 and ESR2, using topological parameter degree greater or equal to 15. In this analysis, we have found the corresponding proteins encoded by common DEGs between AD and PD. Moreover, we also performed DEGs-TF interaction to identify several transcriptional regulation factors, i.e., TFS, that play a vital role in the functions of these common DEGs. We have found 11 significant transcription factors: FOXC1, GATA2, YY1, TFAP2A, E2F1, FOXL1, NFIC, NFKB1, TP53, USF2 and CREB1. At last, we analyzed the known protein-drug interactions to mark out candidate drugs that may have the capability to influence PD and AD. Thus, we identified novel putative links between pathological processes between AD and PD, and possible gene and mechanistic expression links between them. The present study also provides insights into biomolecules and possible new avenues for therapeutic interventions, in addition to the potential biomarker discovery for AD and PD. Conclusions. In our study, using bioinformatics approach, we analyzed gene expression transcriptomics profiles to reveal potential biomarkers that may explain important pathobiological mechanisms underlying AD and PD. We also identified 15 significant hub proteins: EGFR, JAK2, MAPK11, EIF3B, WASL, BCL2L1, CDH1, MCM5, RAN, NCOA3, TBL1X, RARA, RHG12, NCOA2 and ESR2 besides significant molecular pathways and gene ontology terms. TFS that influence expression in the common DEGs was also identified. From protein-drug interaction networks, a number of compounds were identified that might be able to block processes important to AD and PD. In sum, we have explored potential common biomarker signatures for AD and PD that may elicit new aspects of development and progression in these diseases.